Okay, let's start with today's last lecture. And so I know you've all reached, so at least the people coming from France probably are just reaching the peak of their jet lag, but I'm gonna try to keep you awake by also maybe asking you a couple of questions throughout. And I'm hopeful that you will also ask me questions as soon as you have any, or stop me if you get lost, or just interrupt me whenever you think it's a good idea. And so I'm going to talk about epistasis and fitness landscapes, and while making the talk and while seeing like the audience and hearing about things today, I also added like on adaptation, because a big part will also just be on the process of adaptation. I'm a theoretical evolutionary biologist, and I come from classical population genetics. And since we're on the first day, and we've already discovered in the discussion today that there are quite a lot of different ways of thinking about, I don't know, evolution and biology in general that are kind of composed here in the groups. I'm gonna just give you a little bit of a definition here again, and also I call it the high-speed introduction to population genetics, which I'm going to give you at the very beginning now of my talk, but that's not what most of my talk is going to be about. I hope it's not going to be too high speed, so please interrupt me if it becomes like this. So evolution is the change in heritable trait frequencies of a population over time, and there you can, like the trait in question can be any kind of thing. It can be a phenotype, like something you can actually observe, but it can also just be a mutation. And it's not necessary that this mutation has an effect per se on a phenotype and so on. And so we can look at the evolutionary forces that are going to act, and the evolutionary processes, and there are 4 of them, which the main ones, which are mutation, genetic drift, migration, and selection. And so mutation is what creates the variation that you need in the beginning in order to have like heritable trait frequencies and in order to get variation in your population. Genetic drift is the random fluctuation of any traits that you have segregating in the population. Migration is the flow of genes that you can receive from other places. And a selection, we've already heard about, quite a lot about this, is kind of a directional force that might promote, increase the reproductive success of some individuals and decrease the one of others. And so just to get us started here, I would like you to now think about what your favorite evolutionary force is and why. And so by favorite, this can be pretty much anything, like what would you like to learn more about, what do you think is the most important, whatsoever. And I'm gonna first give you 1 minute to think about what your favorite force is. I'm gonna set a timer so we're not gonna get lost here. You have 1 minute from now. Also think about the why, because you're going to have to defend your choice afterwards. 20 seconds. So let's do a vote. So who votes for mutation? Okay. Genetic drift? Okay. Migration? And selection? Okay. So it seems relatively balanced. So now what I want you to do is to group by 2 people and discuss your choice and come up with a common denominator. So you have to choose one of them. And you're also gonna get 1 minute to do this, and then we're gonna see some arguments why this should be the one. Okay, start now. Don't be shy. Well, if it's 3, it's also no problem. So, but you also need to agree on one reason, right? You already have the same reason why? Okay. You guys are not discussing. Okay. Ooh, why is it not stopping? Okay, so I hope you all have decided now, and I would like to hear, oh, it seems to be a very good discussion. Okay, so there will be more time to discuss during the next 8 days. Let's stop now, and I want to hear just somebody who, are there groups that still decided on mutation? What? Suddenly no more mutation? Okay, interesting. Oh, there. Okay, so please tell me why mutation. Okay. So it provides the foundation to actually get different phenotypes and so. Okay. Does anyone else have anything to add about mutation? So you can kind of look at mutations in a single individual, and you don't need to know the whole population for this, you think? Okay. Yes. Okay, yeah. Did everyone understand? No, probably not, huh? So he said that mutations can happen at an individual level, whereas all these other processes, they are more or less population processes. I hope I summarized that correctly. Okay, so let's go. Anyone decided on genetic drift? Yeah, what? Okay. Yes, I totally agree. It gives the stochasticity brings stochasticity into the system, and I think we ignore that actually quite often, and I think that's an important thing to consider, and it's very exciting, and we're going to hear just a little bit more about this, although, of course, since we're going to talk about fitness landscape, we're also going to talk a lot about selection. So, shows migration. Okay. Who wants to start? Yeah, so what she said, I'm just going to repeat it quickly, is that you can basically change the frequencies by bringing in things from other places, and that's actually an important aspect of how populations might evolve. That also is probably ignored sometimes, I mean, not always, but did you want to add something to this? Well, I guess it depends a little bit on your definition of things, but of course, if you receive a mobile element or like whatever from a different species, then you could consider this in the model as migration, I would say. Okay, that's interesting. And so, what's about selection? Now, don't be shy. There must be some people who chose selection. Yeah. So things can increase because of selection, right? Yes. And so why is that exciting to you? I guess one of the easy choices is that you can get really cool things to evolve, right? Like, I mean, natural selection has probably resulted in like lots of interesting things we see, right? So, and that only happened because selection increased frequencies of things. Anyone else wants to jump in for selection? No? Okay, so I guess then we can go on for now. Yes, I mean, this was kind of a trick question because I'm going to tell you in a few slides that I'm interested in the interaction of evolutionary forces, but I just wanted an idea from you guys what you would choose if you were to choose. Okay, so, well, I just have here like one quote about one reason why selection could be the one. And that says here, although many processes shape evolution, natural selection is special because it creates complex functioning organisms. All other processes tend to degrade what has been built up by natural selection simply because these processes act at random with respect to function. And I think that's an interesting aspect also, because all the other things, they somehow build like a mess, and then natural selection acts to sometimes like to direct things into a functional way. And while I don't necessarily agree that selection is my favorite force, I do think that it somehow stands out in a way, and that's phrased quite nicely here. So now if we, like from a modeling point of view, theoretically selection is very simple, right? And so we can just think of something here, like drawn here, where we have time on the x-axis in generations, and then we introduce a new mutation or like a new trait or something, and then if it's selected, in theory, then it can just increase in frequency and fix in the population. And so just seen separately from all the other forces, it's a very simple thing to describe at first, especially if we ignore anything like epistasis, for example. But then what happens is that, of course, in nature, we will never see this happen because, for example, we have not infinite populations, right? So once we get to finite population sizes, this is exactly what might happen with the same selection coefficient in a population that is 1,000 individuals, and where still the selection coefficient is 10%. And what happened here is that it just got lost because of genetic drift, right? And so what I need to do in order to actually see that this is selected is to run the simulation 10 times, no, 20 times. And in these 20 times that I ran it here, we saw what I pictured before in the deterministic case twice. And so I think one important factor when you want to think like a population geneticist is to always consider that there are these interacting forces, and they might, so not everything that is actually selected for might go to fixation, but alternatively also things that are not selected for might sweep through the population. And so we know from classical population genetics theory that the probability of fixation of a beneficial allele, here called U, is this expression, which is dependent on the population size, which is here the large N, and then the selection coefficient, which is small s, and P is the frequency at which this allele is introduced. And that's a kind of a very like classical theory thing. And now if you consider a new mutation that comes into the population just through mutation, then of course so in a diploid population this comes in at a frequency of 1/2n because it appears in one chromosome, and then this thing here simplifies such that you can come up with an approximation that says that the probability of fixation will be just twice the selection coefficient. So if we go back to this example here, then this means that in this population, in a large population, so actually, I mean, if you want to, I can write it down here. So in a large population, divided by 1 to the -4. Okay, so if we introduce that we want to have one copy of the mutation at the very beginning, then we can get rid of this here. And so what we get is 1 minus e to the minus 2s divided by 1 minus e to the minus 4 ns. And now if we assume that this here is large, then we can just get rid of this. And so what comes out is that so this here is roughly 2s. And so the fixation probability of this allele here will be just twice the selection coefficient. And that's actually quite surprising, right? Because you would think that something that's as strongly selected as this thing here would really take off, but it's not really true. And I think that's always important to keep in mind when you think of population genetics and how selected things might actually not sweep it through the population. And that also has a very different and very also a major implication, which is that we also know from coalescence theory that the probability of fixation of a neutral allele, one that has no a selection coefficient of 0 is just the 1/2n if it's introduced in a single copy. And so that implies that if the selection coefficient of this allele here is between these levels, so if the selection coefficient is smaller than 1/2n, then actually it behaves just like a neutral allele. And that is gives us population geneticists huge trouble, because it means that things that are selected might look like things that are neutral, and things that are neutral might look like things that are selected. Do you have any questions about this? Oh, yes. So if you see that the probability of fixation is 2s, right, twice the selection coefficient, and if that selection coefficient here turns out to be smaller than 1/2n, then it basically, no, that's weird. Yes. Yes. Then this is exactly like the probability of fixation of a neutral allele. And so if the selection coefficient then is within these limits here, then selected or like slightly beneficial and slightly deleterious alleles will behave just like neutral alleles, and we can't distinguish them from selected ones. And that's actually what Ota proposed in her nearly neutral theory, and it's really one fundamental realization in population genetics that makes things both complicated but also really interesting, right? And that also creates a large dependence on evolution on the population size. And yes. Well, or the interaction of genetic drift and selection. But maybe I'm just a little bit biased. Okay, but things can get even more complicated, right? Because we also have genome structure, and so things find themselves on the same genome, and different mutations can arise. And so, for example, here you just see a picture in which I simulated a genome, again across time, and then we see the allele frequencies of all mutations that are in this genome over time. And actually, they are colored by the genome position, I think. No, maybe not. But they are colored by, these are all different mutations. And there's one mutation in here that is beneficial. And so you might guess that it's one of these, but you can't really tell, right? And that is because this one mutation that is beneficial, it appeared on the it appeared on a haplotype, and we're here talking about an asexual population in that simulation here, that already had another mutation that might have been, for example, neutral. And that is what we call hitchhiking, because now, and I think actually in this simulation, the neutral hitchhiker was just hanging out as a neutral allele in the population. It was picked up by the beneficial mutation, and then it took off and went to fixation. And now if I showed you this population here, you might see this population, this mutation fixed, and you might say, oh yes, this mutation was surely under positive selection. How else would it have fixed so quickly? But no, that's not actually true. It just took a ride on this other beneficial mutation. And well, of course, we can make it kind of many things that can be, we can make it more difficult by, for example, adding population structure, by adding epistasis, or like your favorite other confounding factor that might happen in real populations. And so I guess this whole thing for our, well, I mean, firstly for the purpose of my talk, but also in general, it's important to keep in mind that selection operates on these like phenotypic differences, and not on a specific like mutation or genotype or so. It will always come with a whole individual, right? And so that makes it so hard to distinguish between these things. Okay, so just to summarize my my high-speed introduction to population genetics, what we as population geneticists usually want to know is how many and which mutations were beneficial when we see like a genome or like when we get population samples. Then we would like to know in general what the proportions of beneficial, neutral, and deleterious mutations are. They're both that appear in the population, but also that fixed throughout evolution. And what is the relative role of selection and the other evolutionary forces in shaping genomes as we see them. And then, of course, we would really like to develop the best methods to infer like the contribution of selection to molecular evolution. And so, yes, are there any questions until here? No? Good. Okay, so that brings me to what we do in my lab. My lab is called Evolutionary Dynamics, and what we study is how populations adapt to challenging environments. And one of the questions that we address is how drug resistance, for example, can evolve, because now after this short, like kind of high-speed introduction, you know that it's almost like you can't really avoid evolution, right? As soon as you get mutations and you get drift to happen and you have variation, there will be evolution happening. You can't really stop it. And then what we would also like to know is which processes drive speciation and diversification in general. And now here my favorite question, what is the role of interactions in evolution? And interactions come both as genetic interactions, but also as interactions of evolutionary forces, interactions of genomes and the environment, and all these kinds of things. So the great thing is that as a theoretician, I can make it very simple, right? Because, for example, what I can do is to say, oh, okay, today I just want to study selection, right? So I'm just going to ignore every other process and build models that only include selection, and then I can really look into what the contribution of selection to a specific scenario might be. And so that's what we do in the lab. So we study evolutionary processes using simple models, sometimes not so simple models, but on average we try to be as simple as possible. We evaluate these models using empirical and simulated data, and we also use modeling to inform experimental design in collaborations with experimental people. So, and I told you that I study how populations adapt to challenging environments. So let me just tell you my definition of adaptation that I'm going to use today. So what I will usually talk about is adaptation as a process of increasing fitness of a population in a given environment. And that is different from how other people talk about adaptation when they especially talk about an adaptation or an adaptive trait. And that is really a trait that has increases its carrier's fitness in a specific environment, and for which you have shown that it has spread because of the direct action of natural selection for its function. And you can already see by these definitions that these are actually quite different, and it is much harder to show that something is an adaptation than it is to say that things adapt, right? And so I think one really nice example where people actually showed that there is a clear adaptation is in these puppet mice here, which have adapted to kind of black stones or like and lighter sand or lighter environments. And what they saw is that actually there's really direct selection acting here on the phenotype of light or dark color. And they showed this by building little models of these and putting them in the good and in the bad environment. And then they looked how many birds had actually hit those models. And so they counted how many of the models were hurt. And by doing so, they could really show that especially this phenotype was under selection in the different environments. And then they also went on later to find like the underlying genetic basis of this. So now let's go back to what do we want to know about adaptation? And some of this overlaps with what we want to know as population geneticists in general. So one thing we would like to know is how big or small are adaptive steps. We would like to know these proportions of beneficial, neutral, and deleterious mutations that I already talked about. We would like to know how mutational effects change depending on the environment. which is already like going to this mouse example, right? Because there's like some mutations that might actually change your phenotype and make you well adapted, and those clearly change their effect if you're, depending on whether you're born in a white or a dark environment, but others might just be the same depending on these two. And how do mutational effects change depending on the genetic background they find themselves in? And that leads to the question that we're going to address also during this lecture, which is what is the role of epistasis? And finally, we would like to know the relative roles of selection and other evolutionary processes. And ideally, we would like to infer this contribution of selection. Okay, so let's talk about, oh, what's happening here? Okay, so let's talk about what we expect adaptation to be like theoretically. So I'm going to go back now almost 100 years and introduce some models of adaptation that people have come up with, and that we can now, using experimental data, we can actually test or evaluate these models. So, two models of adaptation. So this is Ronald Fisher here, and he proposed in 1930, he proposed Fisher's, what's now called Fisher's geometric model. So he said, okay, we have some multidimensional phenotype space, and in this case here, I'm showing it as a two-dimensional one. And our current population resides in this current phenotype, which is the black dot in this picture here. And there's also a fitness optimum, like a phenotypic optimum in the phenotype space. And then he said, okay, mutations happen randomly, and they happen as some kind of distribution of arrows that move you in the phenotype space. And any arrow that gets you closer to the optimum is a beneficial mutation, and any arrow that gets you further away from the optimum will be deleterious. Like, for example, this one here, it carries you further away from the optimum, and so it would be a deleterious mutation where the distance that it brings you further away will be the selection coefficient of that mutation. And people have come up with like really dozens of different hypotheses that derive from Fisher's geometric models in different ways. But some of them are, for example, that if you have a more challenging environment, meaning that you're further away from the phenotypic optimum, you will have more beneficial mutations. That is because if you move the optimum further away, the circle becomes bigger. And so just for geometric reasons, you will have more arrows that actually point inwards regarding to the circle. And so the general philosophy of Fisher's theory was based on the assumption that you have very large populations. So if you read Fisher's book, I think he talks sometimes about like billions or so of individuals. And he also, this is very clearly quite based, like especially if you think of this basic model, based on the assumption that there is a single fitness optimum. Now, in comparison to that, almost around the same time, and not without some kind of clashes between views, Wright also proposed a model that involves like what we call like an adaptive or a fitness landscape. And so here's a picture from his paper, and I think you've already seen one earlier today, where he said, okay, let's assume our fitness landscape is a topographic map where you have peaks and valleys. And so basically, there could be a rugged fitness landscape with many fitness peaks. And now the thing is that the population somehow will move to the closest fitness peak, and so it can get stuck. And so he invoked that you really need small and structured populations in order to get either drift or migration to help you cross fitness valleys and adapt further after getting stuck in a local optimum. And so that's like a very different philosophy. And so that's why I'm going to ask you now, which team are you on? So what do you believe is the mode by which populations often adapt? Are you going to be in Team Fisher, or are you going to be in Team Wright, and why is that so? And I'm going to give you 2 minutes again to discuss, please, in pairs, and decide on one. Okay. So let's do a vote first. Who is on Team Fisher? Yay! And who's on Team Wright? Okay, I feel like not everyone is showing their hands. So again, who's on Team Fisher? Okay, so what is it like? Can anyone phrase like? Are you able to phrase the question about this? Okay, so I think the difference is you have like this topographic map in which populations are moving around. You have a lot of fitness peaks potentially, and so what happens is that if you move here with your population, you will be at a high fitness point, but you might not get away from it, right? Like if you are like in your Fisherian world, then you're stuck at the fitness peak because you can't get any better locally. You would have like a really need a super large mutation in order to get to a different place. And that's why he said you need either very small populations such that there is a lot of genetic drift. And as we said, genetic drift kind of, somehow smoothes the whole thing such that you could suddenly just by having small populations or bottlenecks, for example, that reduce the population size for a short time, you can then weaken the effect of selection and jump to a new fitness peak. Or alternatively, just a second, alternatively, you could get migration where you have one population that hangs out in one peak and then another population that hangs out in the other, and then they can kind of have migration between them such that you can then create like both, you can pass between the two peaks, but you can also get like hybrids and they might have a new opportunity to evolve to a new fitness peak. Does that make sense? Okay, up there. So, well, I mean, the time scale is a different one, right? Where if you have a very large population, you will climb this hill very quickly, and then, of course, you will never be exactly at the peak because you always have mutations, so it will be a cloud that's somewhere around this peak. But in Wright's picture, you have like things spread all over the fitness landscape, and then I think, I'm not exactly sure, but he also, I think he invoked like changes in the environment where the fitness landscape might also change. Yes. At the very beginning, you won't just be spread all over, right? You have to spread somewhere. And so if you start here, even if you start with a diverse population, some might go here, some might go here. But then if that is your very optimal fitness point peak, you might not get there even in 1 million generations. And I think that's the point of the Wrightian model where he says, okay, now if we have small populations and if we allow for migration between small subpopulations, then we can actually, and I don't have the picture here, but he drew these populations in there, and he said, so now if you get migration, then you can get, for example, between here and here, you make a hybrid which gets you there, and maybe it will help you get to the best fitness. Yes, I think, I mean, these things clearly complicate the whole thing, and people have also studied Fisher's geometric model with like moving optima, but for now we're going to consider that we have just one environment, and in one environment you will have one fitness peak. And you also had a question earlier. Ah, yes. So you asked whether the optimum should be a saddle point or a real fitness. If it was a saddle point, and if you assume that you can get mutations in all directions, then you will just by neutral mutations, you will eventually pass the saddle point, right? Yes, I mean, for like, let's not invoke environmental changes for now, right? And there is also a lot of theory about how you want your fitness landscape to look like in order to be most ready for environmental changes, for example. But then, yeah, that's a very good question, but I'm not going to address it for now. Okay, but back to Team Fisher and Team Wright. Now, don't distract me anymore. Let me know which team you are on. Who's on Team Fisher? Okay, so there are a few people on Team Fisher. Who's on Team Wright? I don't think everyone has joined a team yet, but okay. Oh, well, no, I mean, he considered this as the trait space, right? So this would be one trait, this would be another, and then you can add like as many traits as you want. And in fact, what he was considering was a lot of traits, and also that you would start kind of mostly like far away, so you can really go in all kinds of directions and so on. No, yes, I mean, I don't think like so the thing is that this model is just like a continuous model, and it assumes that you have kind of large population sizes such that the fixation probability is actually relative to the selection coefficient. Because otherwise, like you won't even get to the optimum, because at some point you're kind of beyond this threshold of near neutrality, right? No, in one individual it wouldn't work, because you would just fix deleterious mutations all the time. Because, you know, like the thing, that's actually why I drew it like this, because so this darker blue circle here would, for example, be the effectively selected circle, right? But if you move from here to here, then you're kind of in this ring, and that ring is kind of neutral, right? And so the smaller the population size gets, the bigger this ring here gets, and that kind of follows back to what I was talking about earlier with the nearly neutral thing. So even if you move here, and if you have a smaller population size such that the light blue ring is wider, then you're still like, it's still a neutral mutation, and then the smaller the population size is, the more likely you're going to fix this one. So I think it's not considered in the original Fisher's geometric model, and I think it's actually a good point which we're going to address once we look at these little exercises that I've prepared for the discussion sessions, because there we've actually we've created like a fitness landscape that is continuous like this one, but we're evolving it with a specific population size, and then what you can see is how the population size actually affects then moving on that fitness landscape. And you can just try it out by yourself and see the different relationships. I must admit that I don't remember how exactly, yeah, I should know this by heart, but currently I don't remember how it's set up in the original Fisher's geometric. So yes, I mean, here I'm thinking, so this is basically drawn as if we would put one clone kind of in this new environment, and then we would just let it evolve, and then what happens is that you get these mutations. And so, of course, you get many of them, and they will compete, but then with a probability that's relative to the selection coefficient, they will fix, and that way you will get something that we call an adaptive walk on the fitness landscape. And that will get you closer to the optimum with probably successively smaller steps depending on the type of fitness landscape. Does that answer your question? Okay, great. So let's go on. Yes, so this is going to address a couple of different questions, because, for example, from Fisher's geometric model, we can derive things like the distribution of fitness effects, and that will give us an estimate of what the proportions of beneficial, neutral, and deleterious mutations will be. And so it basically leads to this question: what is the shape of the distribution of fitness effects? And I might call this DFE in the following. Just don't get confused. I'm going to try to not do it, but DFE will be the short version. Okay, so people have tried to use just classical population genetic inference methods to see what kind of selection coefficients beneficial mutations have. And if you look at just a couple of examples here, these are not very, like, I mean, these really range over different orders of magnitudes. I think these are all in Drosophila. And so that really didn't, I mean, I think these methods are now becoming much more and more powerful. But if you look at just a sample of genomes from a population, it's very hard to estimate what the probable selection coefficients were that were underlying these different mutations. And then we have some direct estimates, like in another mouse example here. These are like little deer mice. and there where they put these models, and they saw that the selection coefficient of this known phenotype was on the order of 10%. But still, that doesn't give us any idea of what generally new mutations, which effects new mutations would have. So the great thing is that nowadays we can use different types of experimental approaches in order to get a full shape of the distributions of fitness effects. And that's where my collaborators come in. And I was very lucky to kind of get into this group of people when I was looking for a postdoc. So this is my postdoc advisor, Jeff. And then this is Dan Bolon, who developed the method that I'm going to talk about. And Ryan Heatpass, his former PhD student, who was actually responsible for all the data from this experiment that I'm going to show you today and probably also tomorrow. So what they did was they created these engineered mutations, point mutant libraries on plasmids where they can really introduce any possible mutations that they would like to test. And then these plasmids are transformed into yeast cells. And the way it is done is so you create this library of mutants. And this is possible because even, for example, if you have a stop codon here, which would give you a very low fitness or a very deleterious effect, then what is happening is that you have a second copy of the gene in question that is actually functional. And but it's also engineered such that it's regulatable depending on the environmental conditions. So you can basically put these guys here in, and then you can amplify your library such that you get really hundreds and thousands of copies of each of the different mutants, which are then grown together in flasks over time. And what you can do then is to do deep sequencing at different time points. And by doing so, you can get the frequencies of these hundreds of mutants over time, which you can then use to estimate the specific selection coefficients of each of the individual mutants. And so what's kind of cool about this approach is that you get this very systematic high throughput sampling of hundreds of chosen mutations. And in particular, you can also screen mutations that are strongly deleterious. Because if you think of kind of taking a genome from a natural population, you will not see anything that kills you, right? Because it has been selected against no matter how small the population was. So that way we can actually screen the whole distribution of fitness effects that ranges from something that really is lethal to the organism to things that are beneficial. And so it's unbiased. And then because we have these book competitions in the lab, all of the hundreds of mutants are grown in the same environment. And so the environment is really like as similar to them as it gets. And because the experiment is run over a very short time frame, the probability that secondary mutations will appear is very low. And even if they would appear, they would probably not in that time really affect how the frequencies of those chosen mutations are changing. And so there's really a minimized potential for measuring anything but the effects of those single mutations or whatever mutations we chose. And so in a way, and I mean this is wrong, given the definition I've given you, is almost like an evolution-free snapshot of the distribution of fitness effects in the sense that it completely gets rid of any mutation, like new mutation, and it pretty much gets rid of genetic drift because the population sizes are very large, and well, clearly no migration happening because we're in a book competition. And so we can, even though allele frequencies are changing and we're actually using those allele frequency changes in order to estimate selection coefficients, it's not an evolution experiment per se. Okay, so now we can look at this from a modeler's standpoint. And so that's what I did during my postdoc because I also wanted to get an estimate of how accurate our measurements actually are for these experiments. And so what you can imagine to be happening in this population of mutants is that you have like a timeline here and a population size of individual mutant types on the y-axis, and then what you create is this library of the different mutants that are grown together, and each mutant has a specific initial population size, which we want to estimate. And then also what we're most interested in is the mutant-specific individual growth rate. And so each mutant here has also a different individual growth rate, and we can now estimate this by taking these samples of the relative frequencies, and that's basically what the sequencing does, right? So it's a multinomial sampling of those different mutants according to the frequencies in the population. And so if we write down this model, then we implemented this as a Monte Carlo Markov chain, and that way we could get estimates of the posterior probabilities of each growth rate. And so it turns out that depending on, of course, the coverage and the number of time points and so on, we can get estimates of fitness effects that are roughly like smaller than 1%, which is a pretty good estimate, especially considering that we have competitions of hundreds of mutations. And so we could go on then to actually look at the distribution of fitness effects for these different mutants. Okay, so now there weren't so many Fisherians, but I'm still going to show you this part here where we tested or evaluated Fisher's geometric model. Okay, so I've already shown you this. And so two of the hypotheses that you can derive from Fisher's geometric model are firstly that if you change the environment, what's probably going to happen is that you relocate the optimum. So you basically change the fitness landscape, and that basically gets back to some of you guys' questions already. So changing the environment will change the fitness landscape, and if the environment is unknown to that population, it's very likely that the optimum is now further away than it used to be. And so if it's further away, I've already mentioned that, then this circle here will become bigger, and so there's a higher potential for beneficial mutations to appear. And secondly, there's some theory that predicts that depending on the number of phenotypic axes and also on the distance to the optimum, then you expect that the distribution of beneficial mutations should have a specific shape, and it should be either bounded or exponential. And so we set out to actually test this using our data. So let's look at what the data are again. So this is the HSP90 protein in yeast, and all I'm going to talk about regards this very small yellow highlighted region here, which is 9 amino acids big, and which was chosen because it contains very, very different amino acids, and some which are like exposed and buried, and so it seemed to be a good place to start this kind of endeavor. And so what we have is the data for 6 different environments, where 30 degrees Celsius is the happy lab environment for those yeasts. And we had a hot environment and a cold environment, and we also had those 3 different temperatures, but with an added stressor, which would be high salt conditions. And so just to give you an idea of how challenging these different environments should be, the growth, of course, so the relative of the growth of the wild type or our reference type, which came from the happy environment as in those different environments, is like this here. So it still grows reasonably well at high temperatures, but already much less in the cold temperature. And then as soon as you add a little bit of salt, it grows really much slower. And so from our Fisher point of view, this kind of indicates that the distance from the optimum should be really much further when we add the salt stresses. And so what we get then is because we have these 9 amino acid regions and every possible codon at each of these positions, we get roughly 560 mutations per environment. And so these are the same 560 mutations, and we can now evaluate their selection coefficient across the 6 different environments and test what the distribution of fitness effects might look like. And so here's just in our standard condition what the distribution of fitness effects looks like. So here's our selection coefficient estimate. And then we have the different, so these are beneficial mutations in blue here, and very deleterious ones, which I have binned at minus 0.5. And then everything here is somewhat deleterious, and the yellow with the light blue indicates mutations that are not really distinguishable from the reference type. And so this is more or less what we would expect when we think of, well, both like the nearly neutral theory, but also what other experimental studies have seen, where you have a lot of mutations that are around neutrality and very little that is beneficial. But then also a second peak, it's like a bimodal distribution where you also have a lot of mutations that are actually very deleterious. Oh, but I mean, the library is created still in the happy environment, and then you create this big like library of mutants, and then you put it into your treatment, which is then one, for example, with high salt. And so at that point, you're just running this for a couple of generations, and what is it, like 20 generations roughly. And so in these 20 generations, you're not expecting mutations to appear that can actually rise to high enough frequencies to affect anything. And so that's why I said it's kind of an evolution-free. So it's more or less like new mutation-free environment, and everything grows kind of very quickly, and it's maintained in an exponential growth phase, more or less. Well, I mean, we both have like replicates of the whole data set, but we also have, I mean, depending on the study, we also sometimes assume that synonymous mutations have the same effect, and then we can also see whether there are like large outliers. Oh yes, we just use flat priors. I mean, of course, we had some expectations what these distributions should look like, but it works quite well with just the flat priors. Okay. So that's basically what we get. So this is this distribution comes from these 560 mutations. Actually, in that paper, it's not. It's based on the amino acid effect.
How you got the data from here? So you have the experimental data, which is the fitness versus the different mutation conditions under the same environment, and then you're running a Monte Carlo and trying to get the posterior distribution
of the growth rates.
Of the growth rates. But you'll have some likelihood function for comparing the model and the data?
Oh, yes. I mean, that was the model that I showed you, right? So
it's a Fisher model.
Exponentially.
Okay. That's it.
Exponential growth for each of them and multinomial sampling at each of the experimental sampling points. Okay. So now if we go back to our Fisher model, this looks still kind of consistent, right? Because here we expect to be somewhat close to the optimum. So that means that most of the mutations are actually deleterious. And just from like looking at it here, you can see that it's kind of consistent with a close optimum and with like Fisher's theory. And nicely, what we also saw is that, for example, if you increase the salt now, then you suddenly get more beneficial mutations to appear. And that would also be consistent with Fisher's geometric model, where now you have moved the optimum further away, and so you have a higher potential to get beneficial mutations. So that was pretty nice to see at that point. And what was also very interesting was that now if we compared the beneficial mutations that we saw under elevated salinity and just looked at their specific effect in the standard environment, then you can see these lines here. And that showed us that there's like many of these beneficial mutations, they actually have a deleterious effect in the standard environment. And that's what we call like the cost of adaptation, which has also been found for a couple of like many mutations in that were like found in nature. Think of the mice, right? And so here it also looks like those beneficials, they tend to have a cost if they are happening in the standard environment. And so secondly, the second hypothesis we set out to test was this one about the beneficial tail of the distribution of fitness effects. And so there's like a lot of, there's a huge body of theory about Fisher's geometric model, and one part of it suggested that if Fisher's geometric model actually captures real distributions of fitness effects, the shape here, or no, or like if this, no. If you look at the beneficial, if you look at the distribution of beneficial effects, then and you look just at the tail of this distribution, then you should get, you should be able to use extreme value theory, and that should tell you something about the general shape of this tail, and that should tell you again, like something about how predictable evolution is. And so this generalized Pareto distribution has one parameter which would tell you something about this tail shape, and three different tail shapes are possible. So the first one here is the Weibull domain, in which the distribution is actually truncated. And you can imagine this very clearly, like if you think of Fisher's geometric model and you're very close to the optimum, then there's only so much further you can get, right? You're already very close to perfect. And so you have to have a distribution of fitness effects that's actually bounded. And so that's consistent with, obviously, then with Fisher's geometric model if the optimum is closed. And that also means that you have a quite limited potential for additional adaptation, right? Because you can only get so far. That's, yeah. And on the other hand, it also means that the different possible beneficial mutations that are still there, they are very similar in effect. So it's rather unpredictable which one will actually take over in the population. So now the second domain is the Gumbel domain, in which we have an exponential tail of the fitness distribution, and that is consistent with Fisher's geometric model if the optimum is far away or if you have very many phenotypic dimensions. And that means that the DFE is already unbounded, but you have a very low probability of getting large effect mutations. And finally, what you can get is a Frechet distribution, a Frechet tail of the distribution, which would mean that you have an unbounded distribution of beneficial effects, and that you have very highly unpredictable mutation effects in terms of size. But probably, since this tail is also kind of it's long but thin, kind of from an empirical point of view, you might hit the same mutation. So it could be that like the identity of the mutation is easier to predict, but the size per se is more difficult to predict. And so now with this data that we had, we could now use estimate the shape of this beneficial tail of the distribution. And so here I'm showing you the different environments, and then this estimated kappa parameter, which tells us the tail type. And then you get these the confidence intervals here from the posterior from sampling from the posterior distribution. And so what we saw, firstly we saw that for most environments it's indistinguishable from an exponential or like Gumbel domain. But then there was one environment that stood out, and that is actually the one that has the cold temperature and salt.
Did you just fit the tail to a specific distribution?
What?
Did you just fill the last part, the tail part, to a specific distribution of your choice, and you're measuring kappa from?
Yes.
Not the rest of the distribution.
Yes, because it's only true for the tail, right? Like this general, like this extreme value theory describes the tail, right? Mm-hmm. So it doesn't describe the whole distribution. Okay, so the one that stood out was the one that has cold temperature and salt. And if you remember what I told you earlier about the different environmental challenges, this was also the environment that stood out because it had the lowest growth rate. I don't know if I'm showing this here. No, unfortunately not. Okay, so that was the one where you really had only 0.3 relative growth rate to the reference type. And so if that had happened only here, we would have not been like so excited about it. But at the same time, we were also studying experimental data from laboratory evolution of influenza to different types of drugs. And we could infer like also a distribution of fitness effects from the mutations that appeared during drug adaptation in influenza. And it's a very different approach, which I'm not gonna go into detail with. But it's kind of also a laboratory experiment, but we actually looked at all the mutations that appeared and swept to and segregated in the population. And in that case, we also had two environments. One was our like standard environment, which was the lab medium that you use in order to grow influenza, which can, I mean, disputable whether this is a natural environment, but it was our kind of standard environment. And then we had the same, but with an added antiviral drug. And so that would clearly be a challenging environment for the virus. And estimating the tail shape again, we found a very similar result where with no drug in our standard environment, we did not find a tail that looked different from the Gumbel domain. But once we looked at the challenging drug environment, we found this heavy-tailed fitness distribution. And so to summarize this part, in response to a novel environmental challenge, we saw that the number and size of beneficial mutations can increase. And we also observed a lot of cost of adaptation. And that was actually in agreement with predictions from Fisher's geometric model when we displaced the optimal. And following severe environmental challenges, the step size of adaptive mutations might be highly unpredictable. And this Fisher type of tail is not consistent with Fisher's geometric model. So let's get back to like WRIGHT'S model here with the fitness landscape. And let's think about what epistasis actually, yeah, no, I don't know. I think it makes actually more sense to stop for today. Just because then we can start with the definitions again, like we did today, and then we can do the other part as a whole. If that's okay with everyone. Probably, especially the jet lag people who won't be too happy. But yeah, I'm happy to take more questions. Yes.
Hang on. I'm confused. It doesn't seem like you said anything against Mr. Wright in these 10 minutes that we've just gone through, because you've only explored the fitness landscape locally. Mm-hmm. And locally, there's presumably one peak and the other directions are not special. If you go to larger regions of the fitness landscape, mm-hmm. you may find a better optimum somewhere, but it may not have been probed in this experiment. Do I interpret this correctly?
Yes, I think you do. Like, I mean, we've only looked at single-step mutations so far, and you're absolutely right. And but we will see other fitness landscapes tomorrow, which are which are maybe like a little more Wrightian.
Okay. So Wright could still be right.
Yes.
Okay. We'll see tomorrow.
So my question is, how can fitness landscape relate with new topics like niche construction, and like not only like using a changing environment, but also an environment that could be constructed by the same organism?
So yes, that's definitely a great question, and it's not the first time I hear this. I mean, this really is a very simplified model, right? I do think that, I don't know, I haven't thought seriously about how to integrate like feedback like within the population into this kind of framework. So some people have thought about kind of changing fitness landscape in a way that like they called it like a seascape, where you have kind of a fluctuating landscape with time. I'm not, maybe I've also seen something about like a co-evolutionary fitness landscape, but I don't remember right now where I saw this or when or so. Yes, but it's a very good question. And I mean, I think we like, so I talked with someone at lunch about these long-term evolution experiments where they found this like niche construction over time, where you have like coexistence of different types, and so it may be a feedback of the environment. And yeah, I mean, these are all things that are happening in nature, and we're just trying from a very simple concept here.
Could I ask a general question? how do you think the shape of the tail that you were talking about might, or in fact the shape of the entire distribution might differ between a complex or at least a large organism and a microorganism?
Mm-hmm. So there were like when this so there's a paper about this where people actually estimated these tail shapes from population genetic data, or they tried to, and they compared different organisms. I don't know how complex they got. I don't remember. I mean, I remember that for yeast, because I compared that actually with their study, they found that the phenotypic complexity with respect to Fisher's geometric model was like 1. so like a one-dimensional trait space seemed to be enough. I do think that they had other organisms where it was more dimensional. But I can tell you tonight if I look up the paper. The problem is that it's really hard to get enough data for this, right? So I mean, for us, it was kind of limiting. You could see that even for one of my environments, the error bars got really huge. And so it's very difficult to get any good estimates of this if you have only few data points, right? You need a lot of beneficial mutations to get an idea of this thing.
Started from, which is selection coefficients in relation to population size and when drift and selection become indistinguishable. Some estimates by paleontologists of selection coefficients in nature are of the order of 0.01 at least, if not larger, over historical and prehistorical times. So does that tell us anything about whether they have indeed spread by selection, or whether drift was a significant effect? Because this is looking back at the data.
Yes, so I'm not sure I understand.
The reported values of selection coefficients based, for example, on body trait sizes from the fossil data are of the order of 0.01, and in some cases even higher. This is after evolution has occurred. So does that in any way tell us that in terms of what actually takes place, it's really these very large selection coefficients that count, whereas for the smaller ones, it gets jumbled up with drift and maybe it doesn't turn out to be effective?
I feel like it's a tough question. And so you think that there were actually like single mutations that had these types of selection coefficients?
There's absolutely no evidence on mutations. It's because these are estimates from traits. So things like body dimensions and so on.
Okay. Well, I guess then it also believes, like it depends a lot on what you think, whether it's like I mean, it's highly polygenic, for example, for body size, right? So then it would point towards like very small selection coefficients individually, no? So I mean, that doesn't mean like even if we found like large selection coefficients, sometimes it doesn't exclude that we would have a lot of drift happening too, right? And especially if we think of bottleneck scenarios, for example. So I don't think that we need to neglect the effect of drift because we see selection, or the other way around.
But you're saying something else interesting now, which is that measured values of selection coefficients after evolution has occurred, you can end up with different numbers depending on whether you're looking at single gene effects or trait changes.
Well, it's not absolutely clear to me how these selection coefficients that you're talking about were inferred. So
these old criteria which were developed by Haldane and others. So you look at body length, log scale it, look at the difference, so which is really the ratio, and divide by the time.
So like a selection differential type of thing. Mhm. I don't know, I haven't thought about it like this, but I'm happy to discuss more.